Well, it's 9 o'clock, so I think I'll begin. Um, Good morning, everyone. It's my pleasure to introduce Amy DeZern from Johns Hopkins. She's the director of the Bone Marrow Failure Clinic and MDS program. She's also an associate professor of oncology at Johns Hopkins. And her presentation today is on clonal hematopoiesis of indeterminate potential and clonal evolution of myeloid disorders. Um, this is particularly important, I think, as we learn more about the frequency of these clonal abnormalities and its predisposition to MDS and leukemia. But I think also one of the reasons that I wanted to invite Amy here was because as we increasingly see patients who have either autologous transplants or autologous recovery after allogeneic transplant, or as we see more and more patients undergoing gene therapy, I think that we have many more opportunities for studying clonal hematopoiesis beyond those over the age of 50 years. Um, I think of all these young patients that I take care of in the pediatric oncology hematology program, and with increasing numbers of patients going through gene therapy and seeing the myeloid malignancies starting to pop up, I think this is going to be a very fruitful area of research coming forward. But Amy, thanks for coming and visiting with us today. There'll be a Zoom audience, as we mentioned before. As people ask questions, please repeat them, and we look forward to the presentation. And thanks for coming out here.
So good morning to everyone. I appreciate those of you who are here, and hello to everyone who perhaps I cannot see but are out there on Zoom. And I look forward to talking about this topic today for you. So clonal hematopoiesis really encompasses quite a lot of things. And this is what I do in my clinical world and a little bit with my lab colleagues. So I have no disclosures related to this today. So I really want to talk to you about clonal hematopoiesis today and understand the role that this plays in a number of other health issues. So this is really quite a broad overview, and I hope will be some good starting points for us to talk a little bit. Either I'm okay to be interrupted or at the end of the talk, and then for us to think about in our own research, both in oncology and throughout. And this really involves understanding the pathophysiology of clonal hematopoiesis and really what we mean when we say that, and then talk about where we're learning about clonal hematopoiesis, certainly in cardiovascular disease. For my solid tumor colleagues, they increasingly are interested in the blood, which tickles me. And then, as John alluded to, some of the ways evolution to myeloid malignancies or other malignancies is quite a problem. So clonal hematopoiesis is certainly not a new term. It's just one that we're much more aware of. And this is a very pedantic definition that the field is currently using. And right now, we think of it as the acquisition of somatic pathogenic variants in the hematopoietic stem and progenitor cells, with the potential for subsequent expansion over time. And I hope some of my data that I show you today and thought-provoking questions will really be, what is that subsequent expansion and why? I don't really have all the answers. I may generate more questions than I do answers today, but it's an exciting time in the field of hematology for this reason. It's also a little bit of alphabet soup. At home, I do a great deal of teaching at the med school and for the fellows. And alphabet soup in oncology and medicine is the way of the world. When I read the notes of younger generations, sometimes I have to think of the acronyms and what they are. And certainly in clonal hematopoiesis, this is where we are. There's CH, which is really the title of the talk. And I'll just remind anyone who doesn't think about stem cells quite as often as I do that all hematopoiesis is clonally derived. But really, this new term is more of a bad term. It's something negative, that clonal hematopoiesis can be adverse. And I hope I can show you where we need to distinguish those two issues. The clonal hematopoiesis of indeterminate potential is something that a good friend and colleague, David Steensma, really coined a few years ago. And we'll talk about variant allele frequency and what it means in terms of that mutation dose. And this is even something I speak about at the bedside with my patients, because I think it has a lot of relevance. We also jokingly, in the hematology clinic, call it clonal hematopoiesis inciting panic, because really the field doesn't quite know what to do with it, and we're trying to learn. Something I'm going to spend less time on, though I wanted to mention for context, is ICUS. This is idiopathic cytopenias of undetermined significance. And the natural history of this is something that's wrapped up in clonality, except for the fact that these patients are thought not to have clonality. And I think as we have deeper and deeper sequencing methods, these definitions will change. Now, ARCH is a little bit of an older term, but really has become quite synonymous with CHIP because this is age-related changes. And as I get older, I realize how many changes there really are. But there's no specific cutoff for this, but it's why you'll see me set the stage with a lot of what goes on in terms of aging. And then, really, where I think John was alluding to and what we're worried about, especially in the myeloid field, are clonal cytopenias of undetermined significance, or CICUS. And this is something that is a rapidly evolving diagnosis in terms of how we couch it, how we monitor it, and what we think the true risk of progression for this is, because it really begs the question, is this actually a malignancy? And that we'll see. So, really, why are we talking about it? I've alluded to it a little bit. My nice introduction did as well. But the truth is, everyone is talking about CHIP. There's even New York Times articles about it today. And so we certainly want to stay relevant. I am a card-carrying hematologist who works in an oncology setting, and we see it the most, but I think that's going to rapidly change. It has a ton of facets for patients who are cared for by other disciplines. It's been fun for me to interact with people like cardiologists who I didn't often talk to about patient care in the same way. Something that is a reality, for better or for worse, in my opinion, is it's increasingly easy to diagnose. I don't think that that's a surprise to any of us who take care of patients, order these tests. But remember, patients are sometimes taking this into their own hands now as well. 23andMe has a commercial now and again that you might have seen. And I think patients bring their own clonal hematopoiesis questions to the clinic more and more. And this is an interesting thing to balance. But to monitor and intervene appropriately, because I do consider myself a clinician first and foremost, I feel like we need to understand it better. And I'll just mention in the context of this, I actually only treat two diseases to the best of my ability, which are myelodysplastic syndrome and aplastic anemia, which are long disorders of clonal hematopoiesis, and I think can teach us something about this topic. So the issue is what initiates a clone. And many of you are probably aware of this, but just for context, the expansion of a mutated hematopoietic stem cell clone is how all of us have worried about, thought about this CHIP, MDS, and AML, at least in the myeloid range. This is the original figure from the CHIP paper that's fairly ubiquitous now. It's colorful and a picture's worth 1,000 words. And when one of those early mutations continues to grow, it contributes to the feature of the disease. And where we are in the field is really trying to understand that polyclonal hematopoiesis that we all have as part of the general population and who we are, how it changes over the course of aging. And you'll hear me mention some more of this alphabet soup over the course of the talk. But as we age, the most common mutations that are coming out in myeloid panels, just in the general CHIP literature, are DNMT3A, TET2, and ASXL1. But even these three mutations are not all created equal, and this is still an area of burgeoning research so that we can learn more. Somatic mutations are truly ubiquitous in all tissues, and I mentioned I'm a hematologist, and I love this picture, right? We have stem cells, then we have the committed progenitors, and then we have the mature cells. And how those mature cells end up in our varied tissues really can be quite different. And certainly we spend a great deal of time focusing on certain ones, like the blood. But I think our colleagues who, if some of you do cardiovascular research in this particular building or solid tumor research, we're learning how to figure out that clonality and if it has clinical implications or not. I would be remiss if I spoke only of somatic mutations, even though that really, by definition, is the acquisition for clonal hematopoiesis part of it. But there's actually long been a germline predisposition known to CHIP as well. We could talk about this for the entirety of the hour, and I certainly won't. But we're learning from older series and then even newer ones that when you look at germline and somatic variants in known myeloid malignancy associated genes, that you can start to get a picture of which may be more adverse and when it happens, if we can understand the natural history, and then which variants are just associated with the development of CHIP. This is an older paper from a colleague in Blood, but specifically the CHIP that was of concern there on the somatic side of things were JAK2, which we've long known as a myeloid malignancy gene, as well as TET2. But following on germline clonal hematopoiesis, we have to also acknowledge and recognize, certainly in such a large Fanconi center as this, that clonal hematopoiesis and inherited bone marrow failure syndromes is also something we've always seen. And what's a little bit different, and I believe that those patients can teach us globally about clonal hematopoiesis, is that they too, like all of us as we age, start with polyclonal hematopoiesis. But as they age, it's a little bit different than the rest of us, perhaps just gaining in years. They have stem cell attrition. And it's one of the first things that we have to think about, the influence of that on clonal hematopoiesis and what happens in that context. I'll come back to that closer to the end. But going back to somatic mutations in hematopoietic tissue, when this was first recognized, those high-risk mutations that I've mentioned of DNMT3A, TET2, and ASXL1 continued to be picked up. And these mutations are unvarying in the literature in terms of what is the definition of a mutation with CHIP. But just because you can find one of these doesn't mean they're necessarily all high risk. And it's fish mouth plots like this, which really we need to understand who ends up in the yellow and the gray and what is the triggering event at this point where the blue begins to spread out. And we have to think about somatic mosaicism, the clonal hematopoiesis that's gaining dominance, and then certainly the cancer. And I believe that is a question of increased stem cell fitness. And this is a question to which I don't have a great answer for you guys today, because I think the field has some thoughts and some ideas of what allows certain mutations to be higher risk and certain clones to gain that fitness. But it really is the survival of the fittest. And I'll just mention across the literature, it can be quite a challenge to understand what might become the fittest, because the prevalence of some of these mutations is a little bit different depending on the method with which you use to monitor the clonal hematopoiesis. Whole genome sequencing picks up something different than targeted panels. And at the bedside, I am not advocating that everyone get whole genome on any level at this point, but it's a really complicated situation to figure this out. And the relative expansion of those individual clones really depends on what those somatic alterations are in the key genes and how we're figuring out this happens as patients age, and if we are doing things to them that changes that stem cell fitness. Right now, many people broadly think of clonal hematopoiesis only as a surrogate for aging and other diseases, and I'm not sure this is wrong. This is from something a colleague and I did at ASH in 2021, just December, about that increased fitness, and I've already mentioned the genes that are the most prominent. But we really need to understand the extrinsic stressors that we as human beings and our patients experience. And in the aplastic anemia world, certainly the T cell attack at the level of the CD34 progenitor is well known. And I think this is a good example. But that has a different flavor of mutations than perhaps those who have intrinsic defects, like our patients with inherited marrow failure, where there might be normal fitness, and we have to figure out the difference between acquired mutations and that germline defect. And all of this happens over time. And just if you are all researchers, I don't say this to be negative, but both in the lab, things happen at one pace, and then in the human being, things happen at a different pace. And I think that's one of the reasons the field is struggling to figure out how to follow these patients more concretely. So what are the clinical consequences of clonal hematopoiesis? You can see many plots like this throughout the literature, looking at age on the x-axis and the frequency of mutations that are seen. The top panel here was one of the earliest publications in the New England Journal from Sid Jaiswal and colleagues, and it really shows what we probably all know, that clonal hematopoiesis is that surrogate for aging that I've mentioned. And as you increase in decades, you see more and more mutations. And this is why I think patients are taking this into their own hands. People are living longer and trying to understand if this matters. But the mutational profiles are different at the different ages. And here on the right from Mirnal Patnick and colleagues just this year, I wanted to show you what I thought was a very nice plot that they looked at all the largest series in the literature and what the prevalence of that clonal hematopoiesis was. And you can see there's different methods of defining it, at least genetically, whole exome, whole genome, and then targeted panels, which is what we still use at the bedside, and trying to understand what we think the true frequency is. So there's a lot of questions in the literature still. But what has been consistent, and I think the reason I called it clonal hematopoiesis inciting panic is several series now have shown that the presence of clonal hematopoiesis, presumably aberrant, increases all-cause mortality. And that is actually true at the spectrum of ages. Obviously, it's a bit sooner if you are over the age of 70, but it still happens if you're younger than 70. And it's the red line that are mutated patients. And you can see this is a curve that does have a clear trajectory, and it was statistically significantly different for survival in this particular paper. And when you think about other ways to stratify, even when you stratify it by what are called whole genome signature outlier status versus CHIP, that this was a complicated analysis, but again, one of the earlier papers by Zink and colleagues in Blood that defined a baseline as subjects who were neither outliers nor carriers of any mosaic somatic mutations. And then they looked at hazard ratios for all-cause mortality. And the reason I show you this is because these were adjusted, whereas the others were not. So if you look at the top panel here, this was adjusted for the age of the blood draw, as well as how old, the year that they were born, whether they are male or female, whether they smoked, and whether or not they had a diagnosis of cancer. And you can see, unfortunately, that the hazard ratios still show that increasing mortality with increasing CHIP. And then if you look at hazard ratios when somebody gets a subsequent hematologic malignancy, also adjusted for the same variables that I mentioned, these continue to be problematic. But I show you this only to ask the question, is all the decreased overall survival in clonal hematopoiesis from cancer? Now, you might think that stands to reason. I think that's what a lot of people think. But for the benefit of giving you a broad overview of clonal hematopoiesis, the reality is that's actually not necessarily the case. Clonal hematopoiesis is thought to be uniformly adverse and may be non-intervenable. That's a very skeptical view, but that's where the field is trying to move away from. But it's been associated with many, many other adverse events. I can't go into all the data for the sake of time, but you see there a list of medical problems that would certainly be followed by subspecialties other than oncologists until you get to the end. And that's why I think it's so important for all of us to appreciate and work towards the understanding of clonal hematopoiesis. I will show you the CHIP-mediated inflammation and cardiovascular disease, because if I'm very honest with all of you, I'd never wanted to be a cardiologist. And I come from a place where many people want to be a cardiologist from the womb. And so I often just didn't emphasize the blood pressure management in my oncology patients and so forth. But manuscripts like this have really made me rethink that and come back to my internist roots. This is another body of work that's come out of several major labs that are studying clonal hematopoiesis. And we really have a good handle on the fact that the coronary artery disease risk is nearly twofold higher in those who have the presence of clonal hematopoiesis, as are early myocardial infarctions. And as I think back over patients I've cared for, I think we've often attributed a cardiovascular event to other things, which may or may not be correct, but it clearly shows that the blood is driving this as well. And that mechanism of action has really been recently elucidated a bit better, that there's that clonal expansion in the marrow, and there's mutant monocytes, and those monocytes traffic to the myocardium. And of course, inflammation is involved. I would be remiss if I didn't acknowledge that throughout this talk. But ultimately, there's disordered cardiac remodeling that's been shown in the lab, both in mice and in some human models, that heart failure develops or worsening. And then those monocytes traffic to the atherosclerotic plaques, and that's when we get the cardiovascular or stroke events. So increasingly, the cardiologists are well aware of this literature, and they're checking for clonal mutations, and then they're sending them over to hematology clinics, at least in the adult world, to understand how we can help. We want to help, but I'm not sure we're quite there yet. But it's something the public is also quite aware of on the adult side for sure. So let's go back a little bit closer to my home in heme malignancies. I've already told you that CHIP can progress to myeloid neoplasms. There's literature on lymphoid neoplasms as well. And something to sort of hit home today, I hope, by the end of our talk, is that all CHIP is not created equal. There's extensive CHIP, call it other things throughout the talk because the field has not settled on it. But the reality is there are patients who have mutations that are higher risk and burden of mutations that are higher risk to get these cancers, lymphoid, or more commonly, myeloid. And that's shown out in a number of series in a very basic way. This was a particularly large cohort, which is why I've shown you several slides from this manuscript, but it was 17,000 patients that were unselected for any cancer. And when a marker of clonal hematopoiesis was present, there was an increased risk of developing a hematologic malignancy. And that risk is thought to be almost 1% per year. And you can see the difference in the curve, and the hazard ratio in this paper was on the high side compared to some broader literature, but 11 for a hazard ratio is the highest that we've seen. And I'll just show you for any adults in the oncologists in the world, this is very akin to something we've known, which was monoclonal gammopathy of undetermined significance, which is a clonal plasma cell. And that has, for many years, I even learned in medical school that that carries a risk for getting multiple myeloma, which is a plasma cell cancer with a very similar rate of transformation. And the same is true of a monoclonal B cell lymphocytosis to get a lymphoid malignancy called CLL. So in some ways, this is not specific to the myeloid field. It's not even that high of a risk, but it's really taken on quite a intense interest in the field. And I think it's because it can affect so many different things, and the risk is what we consider high. So let's move into actual cancers and how the MDS field, which is where I sit most of the time, and we think about how to diagnose these patients with myelodysplastic syndrome. What the myeloid field has not currently settled on is where the separation is between clonal hematopoiesis and overt cancer diagnoses like MDS. This is an algorithm that's a little bit older now, but just looking at the presence of mutations and the presence of bona fide myelodysplastic syndrome with morphologic dysplasia and where we think it should split out. The main reason to mention this particular paper is this is the first time that the term I showed you in the beginning, CCUS, or clonal cytopenias of undetermined significance, came into the literature. And I thought there might be some that see fewer patients. So I just wanted to show you what these sort of reports look like, what we're dealing with for patients who have clonal hematopoiesis, and why this is complicated on a number of levels. But when we send a targeted panel, so I've moved past some of that background data of whole genome, whole exome, but a targeted next generation sequencing panel and myeloid data, and this happens to be from the peripheral blood. I chose it because it's a dramatic example of a real patient. But this is a patient with not quite normal blood counts, no morphologic dysplasia, if you can believe that, in the marrow. And we have good morphologists in our world, who has an unbelievable amount of adverse clonal hematopoiesis that we can pick up in the peripheral blood. And this is the kind of data that we now find readily available to us. We know what the gene is that's mutated. We even can get the amino acid change. And variant allele frequency is the mutation burden that's reported as a percent in these particular patients. So think back to the picture I made with all the circles and the axis of time. More and more of those circles in that picture are turning pink because they're adverse. And this currently would not be considered enough for an overt diagnosis of MDS, but I think we can all agree, even just by the size of the report, that this is bad. And where the field is struggling is for many, many years in MDS clinics, how to make a formal overt diagnosis of myelodysplastic syndrome is incredibly either rigorous or not, depending on your interpretation. But you have to have low blood counts. You have to have morphologic dysplasia in 10% of what our hematopathology colleagues see in the marrow. And for a long time, it said a specific chromosomal abnormality. Now, I haven't been talking about metaphase karyotyping at all in terms of clonality today because I've been focusing on other mutations, but it's really something to think about how this diagnosis is going to change. And as part of the International Working Group on MDS, this is a moving field, and I hope to be able to share with you guys and others that we're going to get away from this more and more. But I often show patients a Venn diagram because I think it's powerful to them to at least understand where they sit. And everybody fears sitting here, though it may be that we're increasingly changing the size of the circles. And CHIP is one thing. You get more mutations and you have CCUS with lower blood counts, and that's where we need to intervene. There's a number of retrospective series of many varieties, just looking at the pattern of these myeloid mutations in different myeloid diseases. And I showed this pie chart simply because it's pretty clear. As I keep mentioning, DNMT3A is common as part of CHIP. It's actually somewhat less common in MDS and a little bit more common in aplastic anemia. Now, I think if this was redone maybe a decade later, we may see changes in the size of the wedges of the pie. But increasingly, the field is trying to understand how to make diagnoses based on these patterns of genetic information and moving away from the issue of morphology. And I love my hematopathology colleagues, but I think this is where we need to go. And this gets to the clinic. Do you need to wait for changes under the microscope, or is a rising burden, a rising piece of this pie enough to clinically intervene? And I think we're almost there. So I showed you on that previous Venn diagram that CCUS and MDS are increasingly closer together. We've seen that this flavor or this definition of aberrant clonal hematopoiesis associated with low blood counts is really very much akin to myelodysplastic syndrome. And you can see that when you look at the breakdown in many large, well-published series across different mutational spectrums, CCUS and MDS are really quite similar. And biologically, that's probably the important point, that the proportion of mutations does increase as you age, but what is most at play in CCUS and MDS continue to be a problem. And as someone who treats a lot of MDS patients and increasingly sees patients referred for clonal hematopoiesis, I have to acknowledge that the survival of those CCUS patients is very comparable to MDS. I've seen it in my own clinic quite a bit, but it's in the literature as well. And yes, there's a 95% chance over 5 years they will develop overt MDS with increased blasts, morphologic changes, or even AML. But all of it is going to progress, and that's why we're looking to intervene earlier. But it's not just the number of mutations. I showed you that example because that patient has so many, but it's also the type of mutations, as I keep sort of hammering on. The risk of progression is much greater when you have 2 or more. So that's one feature that we have to think about with our patients. Again, the more you have, the worse it is. That greater than 3 is the red line on the overall survival curve with an increasing hazard ratio by quantity. And then the type of mutations. There's a lot of series like this trying to understand the interplay of co-mutational patterns. And I think even though we have some wonderful figures like this, we still don't have that fully figured out because it is quite different in those who have CHIP, maybe because they're aging, and those that we can only understand right now because they were diagnosed at the time of their AML diagnosis. I showed you that report really to set the stage for this particular slide, that the variant allele frequency of the mutations is important. And for a long time, my molecular pathology colleagues were unfortunately a wee bit reluctant to report the mutation dose on the results, which go obviously to us as the clinicians, but also the patients. And many commercial labs do not report that. But this is incredibly important. And I don't know if anyone listening is responsible for your reports or how they think about it. But the variant allele frequency clearly matters. And I think we have a lot of really good recent data to support reporting that out. And I follow that quite concretely in my clinic. The incidence of progression when the VAF gets above 10% is very real. And that may come into some diagnostic criteria for CCUS versus MDS. You may remember in the original definition slide that 2% was how CHIP was first reported, but that was chosen somewhat arbitrarily. I don't fault the authors, but there wasn't enough data, 5 and 10 years ago. But the higher your variant allele frequency of any individual mutation, the worse you do. And that is also somewhat different by mutation. You can see that some of these lines have a greater slope than others. And some of the mutations in the CHIP world that are thought to be less adverse, more favorable, like SF3B1, in the CHIP world, if they are rising, that is associated with more overt disease. And we're also learning about different hotspots, even in mutations. You may remember there's the amino acid change, and we have missense or frame shift. This is really getting very detailed, slicing the proverbial pie in many, many ways. But because we have all the technology, we're going to have to figure out clinically what to do with this particular body of data. But as the clonal hematopoiesis growth rate varies, it is increasingly associated with leukemogenesis based on what your mutation pattern is. And something that the Italian group, Luca Malcavati, who's a wonderful colleague and friend, is really leading the field in, is clonal hematopoiesis-like mutations, which is meant to say not as bad perhaps, versus myeloid neoplasm-like clusters. And I think this is where I most agree with where the field is headed. We've talked about DNMT3A. That's common. But things that are more overtly like MDS, like AML, in these papers from the Italian group that they're considered myeloneoplasm-like, they really are the ones I think we have to be more fearful of. And this is a figure summarizing Luca's work that I wrote earlier last year for an editorial, but it's because it really encompasses all the data that we have. We have clinical phenotypes that we must contend with. We have variant allele frequencies we must think about. Of course, dysplasia is always in the background, and the quantity of mutations. And all of this goes into how likely a patient is to have a problem from what is going on in their stem cells. This is from a survival perspective, as well as from an incidence of progression perspective. And those certain higher risk mutations are increasingly becoming clear over the literature. I think we're not there yet, but dysplasia is one thing. DNMT3A and TET2 and ASXL1, which are the most common, are another. But then if you have a splicing mutation, I highlighted SF3B1, but there's also U2AF1, SRSF2. These are the alphabet soup that we're really building towards. IDH1 and 2 mutations are quite uncommon as CHIP, but if you see them, those are more overtly malignant. And certainly I don't think anyone in an oncology center is surprised to hear that TP53 is really most adverse as a tumor suppressor gene. And even in small variant allele frequencies, it probably progresses universally. So how are the clinical side of things going to handle this? And the short answer is we're muddling through. Lachelle Dawn Weeks, who's at Harvard and a colleague and working, she's recently joined the faculty, is working on something for a clonal hematopoiesis risk score. And this is a recursive partitioning to a population of over 11,000 people. They're from the UK Biobank. And to understand this sort of milieu in which we find ourselves in, we're trying to figure out a definition that is clinically actionable and useful in the field for low risk clonal hematopoiesis, intermediate risk clonal hematopoiesis, and high risk hematopoiesis. And for the sake of not stealing her thunder, we don't need to get into what overtly was done in the methodology, but just that there's going to be different flavors of hematopoiesis that are more or less bad. And certainly if it is the high risk and we have that sort of progression or incidence curve for myeloid neoplasms, we're going to want to see if we can intervene and change the natural history as such. And where the field is going, at least in the MDS space, is clonal cytopenias of undetermined significance clinical trials. The oldest one has been open for a couple of years, and you may chuckle or you may not, depending on what you do, but it's vitamin C in TET2. And there's a much more recent building interventional study, and I'm participating in steering committees for 2 more. Even pharma is really interested in trying to understand where we intervene for these, and where we need to think about, is it TET2, which is a fairly ubiquitous mutation, with something that's pretty benign like vitamin C, maybe benign, or do we need to really think about chemotherapy style drugs? And I won't belabor it, but just be aware that as oncologists, increasingly people are going to intervene sooner and sooner. And I think the watch and wait is less where we may be right now. And I'll just, in this portion of the talk, just to let everybody know that there is something called the National Myelodysplastic Syndrome's Natural History Study. I'm the chair with Mikhail Sekris, and really it's been a sort of labor of love, but it's a observational cohort study of cytopenic patients that might have MDS, might get MDS, and they're followed longitudinally for life. This will be the world's largest tissue bank linked with longitudinal clinical and molecular data. We've had it open since 2016. It will close next year, but in terms of accrual, because we're almost there. But I think studies like this that have a very long natural history increasingly will be part of how we understand this disease. So let's talk more about clonal hematopoiesis. I mentioned earlier I just treat a few diseases, and I couldn't give a talk without talking about aplastic anemia. But this is a picture that sometimes I share with patients when we're trying to decide how to treat them. So going back to our beginning of aberrant clonal hematopoiesis, they might be spiky and red. They can be however you think about them. But if you have some sort of problem, in this case, the stress might be the T-cell attack that is the pathophysiology of aplastic anemia, then we have to decide how to intervene. Now, this is greatly simplified here for aplastic anemia. I don't think we can intervene on all clonal hematopoiesis, but bone marrow transplant is a potential cure for aplastic anemia, as is immunosuppressive therapy, which is not a cure. But I think this very simplified paradigm for clonal hematopoiesis, and I am not advocating that we transplant people with clonal hematopoiesis, but these kind of conversations of intense therapy versus something that might be less intense but doesn't totally get rid of the problem is where the field is struggling with how to intervene appropriately for our patients. And it's because we really want to understand this clonal hematopoiesis expansion issue. The more mutant cells you have relative to your normal population, they may be more fit, they may have normal fitness, but as that fraction of clonality increases, we need to figure out what to do for our patients. There's a lot of really elegant computational modeling studies out there. This one was just published in Science about 2 years ago, which looked concretely again in the red line at that DNMT3A, which has really been one of the most studied of clonal hematopoiesis mutations because it's so ubiquitous. And what the authors were looking at was trying to understand the fitness effect per year and the inferred mutation rate. And I so admire my biostatistician colleagues because this is hard, but this is the kind of really smart people we need doing things like this to understand if we have enough data with enough longitudinal follow-up to be able to help these patients understand their progression so we intervene when the toxicity of any therapy we have meets the toxicity of any disease that they have. And this was something I stumbled across in making this talk. Josh Weinstock in bioRxives just about 2 weeks ago sent some of the transomics, the Top Med Consortium data. And what this group is doing, which is really interesting, is that we have a fundamental understanding that these clones are going to expand, and that's adverse to a patient. We know that there's a diverse set of drivers that probably regulate that. But what we don't really know is the factors mediating that expansion. And the reason we don't are kind of the reasons I've showed you already. There's a paucity of large cohorts, even though we're trying with things like the National MDS study. And longitudinal blood sampling, we do it clinically, but in terms of a place to capture that, we don't have that. So to circumvent that limitation, this group has defined something called the passenger approximated clonal expansion rate. And it's similar to the study I just showed you, though I think a little bit more innovative is this is a validated method to infer that clonal expansion rate from a single time point data. And they're calling it the catchy acronym of PACER. But they've looked at over 5,000 patients that had clonal hematopoiesis and are trying to apply this PACER methodology to recapitulate fitness effects due to different known driver mutations. And certainly, I want to see this in the peer reviewed literature. But what they're sort of showing is there's specific promoters that have slower clonal expansion. And I think I want to see more about this. But then there's protective alleles and driver mutations that are worse. And in the lab, as they continue to model this out, I'm hoping it can come to the clinic. So more to come on this, but I was excited to see this starting to come out. What about if there's any actual solid tumor oncologists listening or in the audience? We've known for a long time that cancer therapy shapes clonal hematopoiesis. And this was a particular study led by Kelly Bolton when she was still at Memorial, trying to understand certain clones in cancer. And increasingly, there can be blood contamination of a solid tumor biopsy. And some of the medical oncologists will bring the report down to my clinic in the hematology part of the floor and say, what do I do with this DNMT3A? And that is going to happen more and more and more frequently. And in this particular cohort, which is a really large unselected group from Memorial of solid tumors, they came to several conclusions. And I just mention it because our solid tumor colleagues are aware of it and patients are aware of it, that the mutations are selected differentially. This gets back to that extrinsic stress we can talk about from smoking. I doubt that surprises anybody. Certainly, we've known for a long time that treatment-related myeloid neoplasm from radiation, platinum, and topoisomerase compounds selects for DNA damage. And so we really see that if somebody has a clone in TP53 already, PPM1D or CHEK2, those are more likely to get myeloid neoplasms. And when you can detect the clonal hematopoiesis prior to starting the therapy for the solid tumor, there's really no doubt that that mutation is still going to be there when the patient gets their myeloid cancer. And I think series like this are useful, they're informative, but we really need to sort of figure out how we better identify patients at risk for that evolution to treatment-related myeloid neoplasm for prevention strategies or at least monitoring strategies. And I'm certainly not able to follow all the solid tumor patients that got cytotoxic chemotherapy, but I am increasingly counseling some paying attention to the mutation that was there, following it over time. We've talked about number of mutations and variant allele frequency, and this is increasingly more common, as is checking a CBC, which is not always done at the time of a scan for surveillance of solid tumors. So what about transplant? I'm sort of bringing it full circle to where I spend some of my time. And clonal hematopoiesis that's due to increased stem cell fitness is sort of what transplant is in a nutshell, right? We're taking a set of healthy stem cells, we presume, and putting into the recipient or the patient who needs some help. But what does this really do to those cells in the body? And I'm not going to get into the way we do transplants versus the way some others do, but it is certainly one of these extrinsic stressors that is a really great place to learn from. And our group uses a lot of related donors. We have access to the donor and the recipient pretty carefully, and there's a lot of unrelated donors as well. And this is one of the first series that showed that maybe we need to be thinking about how we choose the donors for their stem cells. Maybe with clonal hematopoiesis, they're going to go through extrinsic stress as the donor transplant differently. This was a study out of Germany that looked at related donors as well. And 16% of the donors in their series had clonal hematopoiesis, defined as we have up until now. But the median variant allele frequency was almost 6%, which is honestly a bit higher than I would have thought it might be. And we could get into all kinds of caveats here about the CHIP prevalence. These were all people that had a first-degree relative that already had a cancer and these sort of things. But clonal hematopoiesis had more chronic GVHD, a trend to higher non-relapse mortality, but lower relapse and the same overall survival, which is very interesting to think about because of a very feared complication if the donor stem cells have clonal hematopoiesis. Remember our picture of the two patients and they go through that extrinsic stress, could that contribute to a second problem in the recipient? And this was a series from Chris Gibson in Boston, who looked at this very question. And then my colleague in Baltimore, Lukasz Gondek, looked at it similarly with Chris, that if you have a transplant and you become greater than 95% chimeric, but you remain cytopenic, is there a reason to suggest that that is a donor-derived leukemia? And we've had 2 cases concretely, as Lukasz published, of clear donor cell leukemia arising from somatic mutations that were retrospectively found to be in the donor, but that expanded through the stress of the transplant in the recipient to give them a second problem. Which brings us to one of the issues that John mentioned in his introduction about how to understand clonal hematopoiesis and some of the newer technologies that we have to treat our patients, which is gene editing. And for the sake of discussion, I've just chosen the sickle cell example because I live in Baltimore where there's a very large sickle cell population trying to understand how to use gene therapy. And the literature is quite varied, though I'll be candid, it is going to be a problem, right? It gets back to how we've been talking about extrinsic stressors and so forth. You have in sickle cell a very specific issue. Oops, 2 dates there, but just a few months ago, this group in JCI reported that there was not increased clonal hematopoiesis in sickle cell if they looked at it by whole genome signature. But the issue is in these gene editing trials when you are really going through a tremendous stress, right? There's peripheral mobilization, there's ex vivo survival, and there's clonal expansion. That, in theory, should be the stress similar to donor cell leukemia and transplant and gene editing. And so the monitoring is very inconsistent. And this is where I think that we have the opportunity to intervene. And the sickle cell community is incredibly attuned to this in so much as after transplant, if a sickle cell patient is not 100% donor, they may have seen hydroxyurea, they've been sickling and having a lot of inflammation over their life. Coming back to what may constitute high-risk CHIP that we must monitor, the TP53 has reared its ugly head in this report from Courtney Fitzhugh from the NIH. And they were able to show in 2 patients, unfortunately, that even if they went back and looked retrospectively prior to transplant in samples, in those who ended up with overt TP53 myeloid neoplasm, they were not 100% donor. This is autologous recovery of some of the sickle cell patients' own cells. They had mutations that were present at diagnosis that unfortunately expanded over time. And it's this paradigm that the field is worried about, that we're trying to learn about. I don't know that we have a lot of great answers, but it's something that's increasingly in the literature that patients are aware of, and we need some consistency in monitoring. In my last few minutes, I want to leave you with a couple other pearls to understand the current literature, because I don't want you to feel glass is half empty when you leave. Perhaps all clonal hematopoiesis is not bad, and there's some suggestions running around to that. So let's go back to that paper I showed you by Chris Gibson, looking at should we choose allogeneic stem cell donors differently. That, among others, references that I list here, shows that clonal hematopoiesis is present in as many as 20% of donors over age 40. I would be a donor over age 40, so that sort of hits home if I had to do that for a patient or a friend. And that's an issue, right? We've shown you this is age-related. Patients are going to need treatments of this nature, and we have to figure out what to do with it. Let's go back to the CHIP definitions and the most common mutations. DNMT3A comes out here again. But donor hematopoiesis does not always, CHIP in a donor does not always end up in that donor cell leukemia that I showed you in the adverse outcomes section of our talk. It's not always deleterious. And this was a series that Lukasz and I did with Chris and Coleman Lindsley in Boston. We had our patients and their patients, so 2 different transplant regimens, but really looking at donor clonal hematopoiesis and trying to understand what was adverse. And ultimately showing that DNMT3A was the most common and that it was more associated with lower relapse, more chronic GVH, which is that graft versus leukemia that we may need, and ultimately improved survival. Now, very candidly, existing studies in this arena don't inform most of our donor choice scenarios, but we do in all donors over age 40 check for clonal hematopoiesis, but we have since stopped excluding DNMT3A carrying donors. But let's say we saw a TP53. Those are the two ends of the spectrum. We would never use that particular donor. And hopefully today I've shown you in broad strokes why. Another very recent observation, but it's now bringing the neurologists into our hematology clinic. This was a plenary, it's plenary number 5 at ASH, looking at another top med cohort, but that patients with clonal hematopoiesis had a reduced risk of Alzheimer's dementia and neuropathologic features in that particular setting. And it was very elegant work, but to summarize briefly, and it's online as a preprint, they're not in the peer-reviewed literature yet, that mutations found in the blood and in the microglia-enriched brain of these chip carriers were present. That really suggests crossing of the blood-brain barrier, but somehow it must attenuate the risk of Alzheimer's disease. So this is how we're learning that all clonal hematopoiesis is not bad. I don't know that we know what to do with it, but it's something to think about. So I'm going to end up talking a little bit about something that this group I think is known for a long time, but the characteristics of clonal hematopoiesis and inherited marrow failure features, and again, why it may not always be adverse, and that's why I think we have to be careful how we think about this. I showed you my really colorful cartoon that I made of looking at stem cell attrition in this particular special group of patients. Now, they have a very different mutational spectrum than I've shown you with our age-related clonal hematopoiesis in part, because DNMT3A, TET2, and ASXL1 are a lot less common. But I think people here probably know a lot about somatic reversion. There's direct reversion and there's indirect reversion. But there's been a number of papers recently looking at the clinical outcome in this setting, specifically if clonal hematopoiesis is bad or not. We see a great number of short telomere syndromes in my center due to Mary Armanios's work. And there's direct reversion, where the DKC mutation, which is quite causative of dyskeratosis congenita, can revert back. And that there's an indirect reversion that Kristen Trantz and my colleague, have had a patient that showed a functional rescue by lengthening of telomeres. I think that this work is still early, and how we apply to the clinic is still pretty much on a research basis. But the point is, we know it happens. And I think that's how the field is still struggling and is a bit confused. And a seminal paper that came out in Nature Medicine in another inherited marrow failure disease, Mirage syndrome or SAMD9L, they also have clonal hematopoiesis. What we thought was really bad clonal hematopoiesis, in this case, monosomy 7. And there's certain cancer mutations in this spectrum, SETBP1. Here, ASXL1 comes out again. And what we're also seeing, at least in the lab and in a large cohort of these very rare diseases, that there are two types of somatic reversion that, again, are not adverse. There's incomplete rescue potential in these patients, or there's even complete rescue potential. And so this uniparental disomy with 7q that can be protective is certainly not something we hope for, but maybe this is a learning opportunity. How do we induce this in patients? And we certainly don't know, but it's where things are going. And I think, coming back to diseases I think about and treat, we're going to learn a lot from our inherited marrow failure patients in the clonal hematopoiesis range, because their outcomes are usually sooner, coming back to that stem cell attrition. And it's just really interesting to think about if there are ways that we can use this carefully, maybe with gene editing, maybe not, but that's how we have to learn and monitor. So you guys have been great today, but hopefully I've shown you that clonal hematopoiesis increases with increasing age, as do many things. It is a risk for mortality. Right now, that's mostly in the cardiovascular arena and the cancer arena. CCUS, though, I believe, is what we're really talking about when we're counseling patients about the clinical implications on the malignant side of clonal hematopoiesis. And that is something you're going to hear a great deal more about in the near term. We've talked about solid tumors. You need to treat them as their initial solid tumor requires, but monitoring is incredibly key and I believe should probably be done in concert with a hematologist. I've shown you that donor clonal hematopoiesis may not always be adverse, but existing studies don't inform who we choose. And not all clonal hematopoiesis is bad and inherited marrow failure as well. And genomic studies can aid in early identification of this high risk somatic alterations. And I think that's where we really need to spend our effort. So, this is a roadmap of how I kind of think about it, that CCUS and MDS are in the continuum of myeloid neoplasms. The main difference is this dysplasia, and I think that's going to go away. And all CHIP is not CCUS. I think that's, if you take nothing else away, I think that is probably the most important thing to translate to patients. There's true population-based prevalence and severity of CCUS is not known, and that's why you're going to see a lot more larger term natural history studies. But even still, CCUS is not binary. It's not you're pregnant or you're not. It's about the number of mutations, what those mutations are, what the mutational variant allele frequency is, but also the intrinsic and extrinsic stressors that a patient has. And clinical trials are moving this field ahead. So I'll leave you with a few questions. What does stay stable and what doesn't? What other conditions are we going to start interacting with more? And is this chip modifiable? What is too much? And I think I'm very careful not to over medicalize it with my patients. Certainly, we want to push forward in the lab and in the clinic, but we are being increasingly more proactive with CCUS. So I'm grateful to you for listening and being here. And I'm very grateful to my many colleagues that do this work with me at home. So thank you. I'm happy to take questions.
If you have questions, please take the microphone.
Anyway, thank you very much, Amy. So how do you overlap familial cancer predisposition syndromes and then finding 20% of the donors had one chip-like mutation, but presumably have no abnormalities? How far do we have to go? Is one of your last lines, we don't over-medicalize chip, but 20% of donors have something that you've identified, and they all have a relative that had a first degree relative, second that had a cancer.
I think there's a lot of questions tied up in what you're asking because there's certainly well identified true germline predisposition cancers, and those families may be different than somebody with a first degree relative in the presence of CHIP. I'll acknowledge I come from a haplo center where we often are moving to cousins or grandchildren, and those aren't a first degree relative, obviously. And if we can check in the appropriate way for CHIP and then monitor thereafter, we often choose a different donor where we can, and usually we can find one. We are in the proband, the patient, and certainly we do targeted true germline predisposition searches if we're going to use a related donor. But to me, that's a little bit of a separate breakpoint than the clonal hematopoiesis that's somatic.
Hi, I'm in the solid tumor world here. Maybe I'm a little off base, but we do a lot of single cell on ovarian cancer, and we're finding a large population of macrophage monocyte cells in there. And of course, in the solid tumor world, you ask, are they M1 or M2? Are they helping the tumor, harming the tumor? But I'm wondering, is any of this clonal hematopoiesis been tracked in terms of phenotypes of the myeloid cells later on? Like, are they more prone to become more M1-like or M2-like if they have certain mutations? Is that?
No, you're asking a beautiful, yes, it is. You're asking a beautiful question. And the short answer is we're probably not completely there yet, but we have some hints. And I actually think this is where the interplay with some of the cardiovascular literature overlies with some of the solid tumors. And for the sake of time, I didn't bring in all the inflammasome discussions. That could be a whole nother hour in itself. But I think you're right. There's going to be helpful and there's going to be harmful. And that's sort of my intent in explaining about somatic mosaicism in the end is we are learning how to put all these pieces together so that we optimize what's good and hopefully minimize what's bad.
And just a side question. So the companies like Caris and Foundation One that report somatic mutations, how many of those do you think are really not tumor related, but are this clonal hematopoiesis?
So that's a really good question. And Foundation One, they have a person who works there that presented an ASH abstract a couple years ago looking at this question. And there's a tremendous amount of blood contamination to the solid tumor. And some of us have provided some fairly proactive feedback to Foundation One in particular, because I don't know if you've seen those reports, but they come back and say, oh, DNMT3A, you're likely to respond to MDS therapy azacitidine. And patients come in with their report and, and then the solid tumor colleague comes running down the hall, but that's not helpful. And so I think that we're learning that that simply is part of aging. And again, that's why I say maybe we're careful where we medicalize it, because that's probably not actionable for that person with an active solid tumor.
Great. We are going to take a question from our virtual audience. Jaime Modiano has a question. Jaime, go ahead and ask your question.
Yeah, can you hear me?
Yes.
Great. Wonderful talk. Thank you. This is really more just a comment that might be thought-provoking for those of you who are in the medical field and don't have a vet school sitting right next door. We published a paper recently in Aging and Cancer documenting that dogs and humans have essentially broken through an evolutionarily determined age barrier. And so a lot of the diseases of aging that we see in people seem to occur in dogs, although they're not precisely parallel because our evolutionary histories are different. But there is evidence that dogs have something that's similar to CH, and some of the mutations that are seen in humans also seem to occur in MDS and potentially other diseases. And we do know that there's some very benign clonal diseases or clonal conditions in dogs, like CD8 clonal lymphocytosis and others. And so just a plug for the potential to look at dogs where the generation time or the lifespan is, on the order of 10 years instead of 80, where some of these questions might actually be answered.
I think that's really helpful. I always multiply my age in dog years by 7, but this could be really useful now, more so than I thought. So thanks for sharing that.
So that was really interesting. I'm also a sort of a solid tumor person. I know nothing about this.
No,
but what's really interesting is you just started out like it's all mutations all the time. It's these mutations. And I'm like, well, who's studying the epigenetic events? Because we know that, like, is it that stress or life stress and say altered cortisol signaling, which is an endocrine disrupting event, does that increase these mutations, or are there epigenetic changes that are reversible that could be targeted at all? Is SBEAK working on that? Because we work on that for solid tumors, right?
No, absolutely. And I don't mean to simplify it, just only for the sake of time, but just so you know, the Myeloid epigenetic meeting was last weekend in Philadelphia. So it's very hot topic this week. But yes, people are absolutely studying that. What we have struggled with from that very specific perspective in the myeloid world is the drugs that are standard, azacitidine and decitabine, which were long ago used in the solid tumor world, have very different effects in different patients based on different mutational patterns of the epigenetic changes that we see at diagnosis, through treatment, and at relapse. And we're struggling to tell the story in a way that we're understanding, probably because we're missing a piece like cortisol. I don't know.
Sure.
But
Okay.
absolutely, people are doing it.
Great. Well, it has a huge cortisol and glucocorticoid signaling has a huge role in say breast cancer stem cell biology. And so I'm just wondering if some of those stem cell biological signaling pathways carry through, to the hematopoietic system, which they probably do, right? Because GR is everywhere, right? Okay. Yeah. Thank you.
Time for one more question. All right, so I'm also a solid tumor person, and I wanted to follow up on Tim Starr's question and your discussion about the Foundation Medicine report. So we've been doing a lot of cell-free DNA genomic profiling and prostate cancer specimens and clinical specimens as well. Should we be blacklisting certain genes from our consideration just based upon their inclusion in this CHIP panel?
So this is a discussion I've had with someone, actually, to tie it all together, my breast cancer cell-free DNA colleagues at Hopkins. And I'm reluctant to blacklist anything because probably if I showed you nothing, it's that we don't know where the field is and that some of these may be helpful, some of them may not. But I think we just have to be really careful to understand the patterns, the co-mutations, and what's growing and what's not. And I think one of the nice things about cell-free DNA is it's pretty easy, relatively speaking, compared to solid tumor biopsy to understand if that's changing. So I don't think we're there to, there's some clear bad ones, which is TP53, ASXL1 frame shifts, but there's a lot that may not be as adverse.
Well, Amy, thanks so much for spending time with us. It's a really interesting talk, and there may be more questions, and so we'll be happy to the rest of the day. Anyway, thank you, everyone.
No.